Good
So,
afternoon, everyone. Okay,
yeah.
let me switch on my screen.
Okay, hi. Good afternoon. So while Dr. Vanicha is preparing her slide, so let me introduce Dr. Vanicha a little bit. So our final talk will be delivered by Dr. Vanicha Vichai. And Dr. Vanicha Vichai obtained her PhD in biochemistry from University of Virginia. Her research interests involve icosanoid biosynthesis pathway in the giant tiger shrimp and discovery of microbial-derived bioactive natural products. And currently, she is the research team leader of bioassay, biosensing, and bioprospecting technology research group at the National Center for Genetic Engineering and Biotechnology, or BIOTEC, Bangkok, Thailand. And today, she will give a talk on the title, Exploring Nature's Treasure Trove for Bioactive Molecules. Ladies and gentlemen, please join me in welcoming Dr. Vanicha Vichai.
Okay. Thank you for the introduction, and I'd like to thank the organizer for giving me this opportunity to share information on our research at BIOTEC Thailand. As the title of my talk suggests, this talk would be probably less intense on the metabolomic analysis, but I would rather give an overview of how we discover and make use of the metabolites, which in generally we call natural products. So I would give a little bit of overview on the discovery of natural products, and also highlight on some technology that we use in natural product research, and also toward the end we'll share with you on the utilization of natural product and how it's relevant to the current issues. And as you know, that we humans have always been relying on the bioresource, as a source of all the necessities in our life for food, clothing, shelter, and medicines. And natural product is really well known for their use in medicines from the prehistoric time. So there is actually evidence from the that the Neanderthal had used natural products as medicine, and there was a first record of medicinal prescription from 2600 BC and describing the use of herbal medicine from written on a Sumerian cuneiform tablet, and it's described different various herbal medicine which we are still using until these days. And later in the 1500 BC, there was a famous Egyptian papyrus paper that lists over 800 plant medicine used for to cure diseases. And fast forward to the 19th century and 20th century, we started to have a more modern discovery of medicines. And there was the first medicine discovered from microorganisms, which we all know, Penicillium fungi, that was discovered in the early 20th centuries. And since the beginning, since the discovery of penicillin, there has been a burst of discoveries of various antibiotics from microorganisms and plants during the 1940s to 1960, which is well known for to be a golden age of antibiotic discovery. So we fast forward to the 21st century. Newman and Craig, have compiled data on the chemical entity that have been approved as a drug by the US FDA, and have been compiling data since 2012 and have keep updating the information, and the last published in 2020. And interestingly, for after we having been searching for natural product as medicines for almost 40 years, natural products remains the most prolific source of new drugs. As you can see from the chart that they show in their paper, various they have categorized chemical entities into various categories, such as biological macromolecules, unaltered natural products, botanical drugs, natural product derivatives, synthetic drugs, and synthetic drugs that have been inspired by the structure of natural products. So from their analysis, over 60% of the small molecule drugs that have been approved by the FDA from 1981 to 2019 are either derived from natural products or inspired by natural products. So there are reasons why natural products are a good source of medicines, because they have a great diversity of chemical structures, and they also, based on the analysis of their physical chemical properties, they occupy the chemical space, which are highly relevant to drug discovery. And since natural product is a product of a very long evolution of living organisms, so they kind of have been optimized by evolution to serve some biological function. And they are generally more readily absorbed than synthetic drug. And also, interestingly, why most synthetic drugs require structural modification to be appropriate use as drugs. Natural products usually can go straight from heat to drugs. Examples of natural products that we are still using or benefit from nowadays include cyclosporin A, which is an immunosuppressant isolated from a kind of fungus. Halichondrin B is a natural product that was isolated from Japanese sponge, but you can see how complex the structures of this molecule is. But it was found that only half of this structure was sufficient, to have biological activity, and its synthetic analog, eribulin, has been approved for the treatment of metastatic breast cancer. And another natural product that has inspired the production of the synthesis of many medicine was mevastatin, which was cholesterol-lowering agent produced by Penicillium fungus. And the discovery of these molecules led to synthesis of several statin drugs, which we are using nowadays, including atorvastatin or Lipitor. So traditionally, natural products are discovered through this kind of steps, starting from collecting the organisms and and then the organism from which we make extract from, and this extract could be fractionated and then subjected to multiple rounds of fractionation, as well as evaluated for biological activities, and back and forth between biological evaluation and fractionation would lead to an identification of single bioactive compounds. But this kind of conventional bioassay guided isolation has been used for almost centuries, and it is still using nowadays. But there's a lot of limitation to this kind of discovery scheme. For example, to have access for the biological materials that are used as sources of natural products require tremendous resources and expertise for sample collection, identification, and this is more complicated by the regulatory, the law and regulations of each country, which are different from each other. And nowadays, it is widely regarded that to collect biological material for natural product bioprospecting, you need to comply with the CBD principle of access and benefit sharing. And another major limitation is the isolation of natural product, because usually most of the time natural products are present only in small quantities in the organism that produce the natural products. And when we extract make a crude extract of the organism, usually the chemical constituents are too complex to be easily isolated. And last but not least, since we people have been screening natural products to identify medicine for decades, there's a trend that you have less and less success in the discovery of new compound, and this kind of scheme usually lead to the rediscovering of known compound, which is considered the most holdback of in the natural product discovery research. And last but not least, the technical aspect of structural elucidation of natural product is very difficult, and usually you require sophisticated equipment and specific expertise in natural product chemistry. So because of these limitations, many pharmaceutical companies that has natural product research program have closed down their this research program, like during the late 1990s and the early 2000s. And but since, however, there are more in natural product research is becoming more interested in the area in academics for because of the advance in the technology that help us to overcome these challenges. And there are several strategies that that researchers in the area utilize to improve the efficiency of natural product discovery. For example, the National Center of Institute of the United States, or NCI, has a program for natural product discovery, in which they created a library of partially purified natural product fractions. And these fractions are provided to research community free of charge. And because of the availability of these fractions, this allows, which are less complex than the crude extract. So this allows the screening of natural products on high throughput screening platforms. And also this helps simplify the isolation of bioactive compounds. And as you can imagine, that this kind of research program require a lot of resources. So it could only be supported by a large public institute that have public funding. Another major strategy, and I think this is probably the most well-developed strategy that used in the natural product research, is the chemical dereplication. The purpose of chemical dereplication is to reduce the redundancy of the natural product discovery, because as you know, the isolation of natural product compound is very time consuming and costly, and it's therefore, if you have a process that can help to identify known compound rapidly early on in the screening process, you can reduce the waste of labor and resources during the isolation steps. So, and this purification process is very important for natural product screening workflow to be efficient. So generally, the natural products purification workflow looks something like this. Probably there are some variation of the technology used, but mainly you would need to be you would have to have the capacity to collect the biological materials and prepare extracts or pre-fractionated extracts, which would then be analyzed by various techniques, and this is generally called metabolite profiling, which also include metabolomics analysis. And most widely used techniques are often combination of different analytical techniques, like with or without separation or chromatographic steps. So these example of hyphenated analytical techniques include TCMS, LCMS, LCMS-MS, or LC-NMR or LC-NMR-MS. So these platforms are often specific, like for each institute or laboratory, and therefore, each institute or laboratory usually generate their own natural product database, which could be used to match against, which can be used for search for the match against the metabolite that we analyze in single extracts. And if you do the search and then discover and find a match between the metabolites in your extracts and the database, so usually these known compounds are discarded from the screening process. If you turn out to find unknown metabolite, you would follow up on the analysis or the isolation of bioactive compounds. So this is just like a general scheme for natural product purifications. But as I mentioned before, like the database are generally quite specific to each platform, and it depends on the availability and quality of spectral libraries. So so a lot of pharmaceutical companies they build this kind of database as their own proprietary, but there are more and more public efforts to comply the database and standardize the format of the data. Another approach that are widely used for the isolate of natural product nowadays is the genome mining. This is we have benefit from the advance of the genomics technology and genome sequencing. So therefore, we have thousands and thousands of genome microbial genome in the database, and we can access this genome to identify the biosynthetic pathway that that would lead to the production of natural products. And since many genome sequences have been available, it becomes obvious that a lot of microorganisms possess greater genetic potential to produce natural product compounds than the natural products that we have been able to detect under laboratory conditions. So by through genome mining, we can increase the access of secondary metabolite biosynthetic pathway that are silent under laboratory conditions. And then this also allow the isolation of natural product compounds that are present in small amount in native strains. So generally, the workflow of the genome mining include the obtaining the genome sequence and then analyze the genome sequence using available software to predict the biosynthetic gene cluster. And then from the biosynthetic gene cluster, we would predict the structure of natural products that the microorganism might be producing, and then we would focus on the gene that that encoded the biosynthetic pathway. So this is like just an overview of the technology that are available to us nowadays. So so I'd like to switch gear to the research, the natural breed product research that we are doing at BIOTEC that address the current problems as especially right now, everyone is focusing on identifying new cure for the COVID-19. And we have a research project that aim to identify inhibitors of coronavirus, and this project is actually initiated by one of my colleagues, Dr. Nopon Shuti Vitunchai from Virology and Cell Technology Laboratory at BIOTEC. And we make use of the natural product collections that the biological extract collections that we have at BIOTEC. Since 2009, we have generated a repository of crude extract from cultures of fungi that were isolated in Thailand. And as you can see, we have accumulated more than 50,000 fungal extract, and these are derived from different kinds of fungi, which are isolated from different ecological systems or different taxonomic groups. And then, from these extract collections, we select like about 2,000 extracts that we know that they are not cytotoxic, and use this as a starting material for our screening. And so the screening system that we use to identify the coronavirus inhibitor is a model using porcine epidemic diarrhea virus, or PEDV, instead of the SARS-CoV-2, which as we all know that in order to work with the COVID-19 virus, we need to have the BSL-3 facility. So to facilitate this screening, we use the PEDV virus, which is a related virus from the related coronavirus. And so based on the rationale that this virus share similarities with SARS-CoV-2 in the non-structural proteins, like for example, polyprotein 1AB, RNA polymerase, and helicase. And so we use this system to screen our fungal extracts. And using high content screening system, Opera Phoenix from PerkinElmer, and we use Bureau cell that has been genetically labeled to express GFP, and use the viral model PEDV that engineered to express mCherry, and using with this system, we use high content screening to analyze. So if you if there is a virus infection, you can see that the cells will form what we call syncytia, which is like a fusion of several cells. And then we can superimpose the image of mCherry imaging and GFP imaging, and then identify the specific syncytia caused by viral infection. And these are this panel just an example of screening results. If you can see that the viral GFP cells are shown as green colors, and the virus labeled by mCherry, this more probably more brownish than red, like different spots. And we detect the cells viability and viral replication at 15 hours and 72 hours post-infection. And then you can compare between the mock infection without virus. There will be the cell will be all green, and there will be no viral syncytia. And with the positive control, nafamostat, which is known coronavirus inhibitor, you can see that at 50-hour post-infection, we can still, the cells are fully viable compared to the control, but at 17-hour post-infection, they are partially destroyed. So, which means this drug cannot inhibit the virus completely. But with the extract that we identify that we use for the screening, for example, extract A, you can see that while at 15-hour post-infection, the virus was still inhibited, but at 22 hours, you started to see some destruction of the cells and the proliferation of the virus, as opposed to extract B, which show the no infection of virus until 72 hours. And for another extracts, we found that cells have been damaged from 15 hours. And so this suggests that this compound might be toxic to the cells. So these are just example of the screening results that we have obtained. And just to show you how we approach with this screening of the inhibitors, starting from 1,000 to 205 fungal extracts, we first screen with the high content screening, and select for sample that can produce 100% inhibition of the virus at 15-hour post-infection, and also at 72 hours post-infection. And then we confirm by second screening, after which we select the hits that has been confirmed from the second screening. And then we determine the half maximum effective concentration of the extract, and then verify the activity. So, so starting from these 1,200 extract, we ended up with 62 hits, and with various ratio of virus from extract from different fungal groups that give the hits. And from this sample, we were able to verify 4 extract that has a stable activity. And right now we are in the process of following up for bioactive compound in these extracts. So this is where we are for this coronavirus in screening. And another area that you may not think that natural product would have much contribution, but they are actually contribute a lot for agriculture in the area of crop protections. As we all know that the problem of health toxic chemical from farming of serious global concerns. And the agrochemical industry are increasingly turned towards the discovery of natural products because they have various advantages and they have been utilized for different functions. So they can either be used directly as natural product in the form of natural product, and also they can be used as can be used for the synthesis of analogs or the synthesis of chemical compounds that have improved activities. And also they can be used to validate the mechanism of actions. And in Thailand, we have various, we have a lot of problem from fungal diseases in agriculture. So due to the limit of time, I'm not going to go into details. But at BIOTEC, we are interested in identifying fungicides from natural product and using this kind of natural product screening workflow where we screen microorganisms using, these include actinomyces and gram-negative bacteria, and we screen them with rapid screening using various fungal pathogens, and then we confirm the activity in liquid cultivation, and then we use a de-replication platform and which include the profiling by LC-DAD and LCMS, and this is carried out in parallel with the bioassay using and microfractionation using UHPLC and DAD, and by combining this microfractionation and chemical profile analysis, we identify the fractions that composed of the possible bioactive compound, and we replicate this the metabolite of extract of interest with the spectral library, which is which we have established from natural product compounds isolated from microorganism, and the construction of the spectral library is still ongoing. And then so far we have not identified a hit that identified as new compound. So eventually we hope that once we have identified promising natural products, we would use the genetic approach to enhance the natural product productions. So I'd like to summarize that for after thousands of years of natural product bioprospecting, they still remain the most promising source of bioactive molecules, which are still relevant to human needs and to the global problems. And as you can see that natural product research demand a lot of investment on biological material sourcings, equipment, and human resource. And but there can still be success in natural product discovery with the continued improvement of technologies and strategies. And last but not least, natural products research require multidisciplinary collaboration. And with this, I'd like to thank my collaborators from the various research team in biotech, including the microbial diversity and utilization, microarray, bioorganic chemistry, enzyme technology, virology and cell technology, and bioassay research team. Okay, thank you.
Thank you very much, Dr. Vanicha. That was a really nice and informative talk. And are there any questions? Particularly for this session. So if not, we can move on to the panel discussion and networking right away.